Welcome back to Power in Pink. My name is Dr. Kinjal Desai, and today I will be talking to you about epigenetics. To understand epigenetics and how it relates to breast cancer, let's start with what we know. The human genome has an estimated 20 to 25,000 genes, and these genes are susceptible to being changed or mutated. When specific genes, called proto-oncogenes, get mutated, it can lead to breast cancer. But what percentage of our DNA is actually genes? It may surprise some of you to learn that the part of the DNA that makes proteins, or what we call genes, is less than 2% of our entire genome. The remaining 98% was previously called junk DNA because very little was known about it. Interestingly, a lot of risk associated with developing breast cancer actually falls in this region, which is what I decided to study for my PhD. Many different labs from around the world have worked together to figure out that this junk DNA actually contains regions that have very important influences on genes. Let's call them non-genes. So, as you know, genetics is the study of genes. Epigenetics is the study of how genes and non-genes interact, and this can be influenced by a number of factors, including the environment. What do we know about non-genes? We now know that non-genes can act in different ways to control genes. They can regulate genes by turning them on, off, or even pausing them until the time is right. During my PhD, we found that non-genes can get mutations instead of the gene itself, which alters the way they control the gene. This causes overactivation of the proto-oncogene, leading to breast cancer. So where do we go from here? Drugs already exist against many of the mutated genes that lead to breast cancer. The next step for researchers is to figure out how to target the non-genes along with the genes for a more effective therapy.